genetic informations that are contained within the DNA, they are then transferred from one generation to the next generation that we can discuss under the heading flow of genetic information. तो इंफॉर्मेशन या इंस्ट्रक्शंस के फ्लो से पहले क्या होता है कि जितना भी जेनेटिक मेकअप है सेल का वो डिवाइड करता है एंड देन इट इज़ ट्रांसफर्ड टू द नेक्स्ट सेल और इवन एट हायर लेवल्स हम ये कह सकते हैं कि इट इज़ ट्रांसफर्ड टू द नेक्स्ट ऑफस्प्रिंग. Then at the molecular level, what happens, especially in the biological system or within the cell, that this flow of information, it is actually within one macromolecule to another macromolecule. For example, the informations are the codes that contain instructions. They are transferred from the DNA to DNA or from DNA to RNA and further on from RNA to protein in the form of functional product. And this concept, it is very famous. As a student of molecular biology, you may be well familiar with this central dogma of molecular biology concept that states that how genetic informations they flow from one macromolecule to the next macromolecule. Here the informations they can be transferred from DNA to DNA by the process of DNA replication, and then from the DNA to RNA by the process which is called as transcription, and then informations from the RNA they can flow back to the DNA, and this process it is very common in some viruses by the process of reverse transcription. And then from RNA the informations they are translated into a functional product which is any protein. Now what happens in eukaryotic cell? Eukaryotic cell, you are familiar that they have well developed, you can say that nucleus that contain the DNA that is membrane bounded. So this is the characteristic feature of eukaryotic cell. So here the information or the genetic informations they are transferred from parent cell to the next daughter cell. And the similar situation it is in prokaryotic cell like the bacteria. So here bacterial cell it can divide its DNA, and then the DNA it is equally segregated into the next daughter cells. And during this transfer of DNA we can manipulate by inserting our gene of interest by using bacterium as a host organism, because this bacterium it is having very short lifespan, rather a very rapid cell division. One cell divide into two within a short span, maybe within 20 to 30 minutes if proper nutrients are available. So we can produce millions of copies of our gene of interest by using bacterium as a host organism.